Dear student, I am Dr. Prakash Mungli, a professor of biochemistry. So in this fatty acid oxidation lecture, I will be explaining you oxidation of odd number carbon fatty acids and also oxidation of very long chain fatty acids. Now let's get into the details. Oxidation of odd number carbon fatty acid. So what is an odd number carbon fatty acid? So it is there in the name, any fatty acid that contains an odd number carbon, can be like 17 carbon fatty acid or 15 carbon fatty acid, 13 carbon fatty acid. So whenever fatty acids like this are undergoing oxidation at that time, so there are certain products that are formed which are little bit different than the oxidation of even number carbon fatty acids that we have already seen. So in the even number carbon saturated fatty acid oxidation, which undergoes a beta oxidation, there will be release of a 2-carbon acetyl-CoA molecule every time. Whereas in the oxidation of odd number carbon fatty acids, similar beta oxidation reactions will be going on starting from the carboxyl carbon. Every time 2 carbons are released as acetyl-CoA in each beta oxidation spiral. But in the last beta oxidation spiral, there will be release of an acetyl-CoA molecule, 2 carbon, and also there will be release of a 3-carbon propionyl-CoA molecule. That's the only difference in the between the oxidation from a saturated even number carbon fatty acid when compared to an oxidation of odd number carbon fatty acids. Okay, that is the one. So the difference is there in the last beta oxidation spiral. Okay, let me show you that here with the figure. So, number 1 carbon, let me just color that here with purple color. Number 1 carbon, we have number 2 carbon, number 3 carbon, like that. Let me just go on numbering here so that you know it is an odd number carbon fatty acid. Number 4, number 5, number 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 and 17. So it's a 17 carbon fatty acid that is shown here, which is an odd number. Okay. Now, oxidation is going on in the matrix of mitochondria. Beta oxidation is the one that goes on. Why beta oxidation? Because there is no problem for conducting beta oxidation, because second carbon is an alpha carbon, third carbon is a beta carbon is free. So beta oxidation can go on. So all these dashed lines that you are seeing here, I am going to take out all these numbers now. So the dashed lines that you are seeing here, it is the indication of beta carbon which is going on between alpha and beta carbon bond. So the second carbon and the third carbon bond between second and third carbon is broken down and there will be release of acetyl-CoA molecule. So this kind of beta oxidation will go on just like a regular beta oxidation that we have seen with even number carbon fatty acid oxidation. So this is number 1 spiral and number 2 spiral, number 3 spiral, number 4, number 5 and number 6. Now the beta oxidations that I have written here, so number 1 to number 6 beta oxidation. So the 6 beta oxidation spirals which are going on there. In each of these beta oxidation spiral, let's look at the acetyl-CoA molecules that are released. So one acetyl-CoA here and the second acetyl-CoA, third acetyl-CoA, fourth acetyl-CoA, fifth acetyl-CoA and sixth acetyl-CoA. So 6 beta oxidation spiral will give you 6 acetyl-CoA molecules, and 6 acetyl-CoAs means each acetyl-CoA is 2 carbons, so 6 multiplied by 2 is 12. So 12 carbons are all released as acetyl-CoA. So 6 acetyl-CoAs are released now. Now at the end of this 6th beta oxidation spiral, so what I will do is I am going to color this and take it out. So we have already released 6 acetyl-CoA molecules. Now I am going to number carbons in the remaining fatty acid here after that 6th beta oxidation spiral. So it will be remaining as C double bond O S CoA activated carbon fatty acid here. So how many carbons in this? Number 1, 2, 3, 4 and 5. this is a 5-carbon fatty acid. Okay, so this 5-carbon fatty acid will undergo one final beta oxidation spiral. So now let's look at where is alpha carbon and where is beta carbon. So alpha carbon is a second carbon, is an alpha carbon, third carbon is a beta carbon. So the beta oxidation is going on over beta carbon and break the bond between alpha and beta carbon. So that is what is shown here with the dashed green line. So this part of the 5-carbon fatty acid is taken out and released as an acetyl-CoA, 2-carbon acetyl-CoA, which has 2 carbon. Out of 5 carbon, 2 carbon removed, and this part of the fatty acid will be removed as a propionyl-CoA. Propionyl-CoA, that is a 3 carbon, and that is what is shown here, propionyl-CoA released. So the point here is at the in the last beta oxidation spiral, that is this 7th beta oxidation spiral here, because already we have done 6th beta oxidation spiral. In the 7th beta oxidation spiral, when 5-carbon fatty acid is remaining, one last time beta oxidation is going on between alpha and beta carbon and release one acetyl-CoA, 2 carbon, and 3 carbon propionyl-CoA release. So that means in oxidation of odd number carbon fatty acid in the very last beta oxidation spiral, so instead of 2 acetyl-CoA molecules, so there will be release of 1 acetyl-CoA and 1 propionyl-CoA. So that means we are going to get at the end of beta oxidation spiral, so we are going to get 7 acetyl-CoA molecules, 7 acetyl-CoA molecules and 1 propionyl-CoA molecules. Okay, 7 acetyl-CoA is 14 carbons. So let me write it down again. So 7 acetyl-CoA, 7 acetyl-CoA means 7 times 2, 14 carbon, and a propionyl-CoA molecule, 1 propionyl-CoA, and propionyl-CoA is a 3 carbon. So 14 plus 3, it is 17 carbon. So that's how we have broken down or oxidized 17 carbon fatty acid into 7 acetyl-CoAs and 1 propionyl-CoA. So all these acetyl-CoAs will go into TCA cycle, which is going on in the matrix of mitochondria. It will undergo TCA cycle. Now what happens to the propionyl-CoA? Propionyl-CoA cannot just directly go into TCA cycle, so it has to undergo further reaction. Now the propionyl-CoA that is released in the previous reaction upon oxidation of odd number carbon fatty acid, in the last spiral, 7th spiral, we get that 3 carbon propionyl-CoA. Now the 3 carbon propionyl-CoA will undergo carboxylation process to become 4 carbon methylmalonyl-CoA. That is what is shown here in the first reaction, and this particular reaction is catalyzed by enzyme propionyl-CoA carboxylase, and this propionyl-CoA carboxylase, it needs biotin as a coenzyme. It is doing carboxylation reaction where ATP is used and ADPs are released. Now once you get a methylmalonyl-CoA, this methylmalonyl-CoA, initially it is a D-methylmalonyl-CoA which is converted into L-methylmalonyl-CoA by methylmalonyl-CoA epimerase enzyme. Not really that important reaction. Now once you get methylmalonyl-CoA, it will be converted into succinyl-CoA. Succinyl-CoA is a 4 carbon molecule. So the 3 carbon propionyl-CoA will be converted into 4 carbon succinyl-CoA. So conversion of methylmalonyl-CoA into succinyl-CoA, it will be done by enzyme methylmalonyl-CoA mutase, and that enzyme needs vitamin B12 as the coenzyme. Specifically, it needs deoxyadenosyl-cobalamin as the coenzyme. Now the succinyl-CoA can go into TCA cycle, and in the TCA cycle, succinyl-CoA is converted to succinate, succinate is converted into fumarate, fumarate into malate. And in the cases like when the person is in fasting condition, if the gluconeogenesis process is going on, if the cell needs glucose, as I have explained to you in gluconeogenesis lecture, so that succinyl-CoA which is going into TCA cycle, becoming succinate, succinate to fumarate, fumarate to malate, malate can move out of mitochondria into cytoplasm converted back into oxaloacetate, phosphoenol pyruvate, and go all the way to glucose. That's how a odd number carbon fatty acid oxidation, which is giving propionyl-CoA, can contribute to glucose formation in gluconeogenesis. That means fatty acids can contribute to glucose formation, but only odd number carbon fatty acid, and the other that can do it is a branched chain fatty acid, which we are going to see later. Alright, so this is how a propionyl-CoA is converted to succinyl-CoA to get into TCA cycle or gluconeogenesis process. So the previous reactions that I have explained, I have written here just as a recap or a review. So propionyl-CoA is converted into methyl-malonyl-CoA by a biotin dependent enzyme, propionyl-CoA carboxylase. And then the methyl-malonyl-CoA is converted to succinyl-CoA by cobalamin dependent enzyme called methyl-malonyl-CoA mutase enzyme. So two enzymes here. One is propionyl-CoA carboxylase, other is methyl-malonyl-CoA mutase. Once we get succinyl-CoA enters into TCA cycle for a further oxidation process, or it can enter into gluconeogenesis to make glucose. And acetyl-CoA is completely oxidized or oxidized to give energy, or in the liver it enters into ketone body formation. That is the other acetyl-CoAs that are coming from oxidation of odd number carbon fatty acids. Like 7 acetyl-CoAs that come, they can go just into beta oxidation or ketone body formation. Propionyl-CoA can go into oxidation in TCA cycle or simply enters into gluconeogenesis. Now let's get into diseases that are associated with oxidation of odd number carbon fatty acid. Our first disease here is propionic acidemia. What is this propionic acidemia? Propionic acidemia, it is an autosomal recessive condition, and it's an organic acid disorder due to defect in propionyl-CoA carboxylase enzyme. Or it can be an acquired disease, like acquired cause for propionic acidemia is because of deficiency of biotin, which can lead to decreased activity of propionyl-CoA carboxylase. So what happens here? There will be toxic buildup of propionic acid, which can be found in the brain, and also it can be found in other body parts. Now, clinical imaging evidence suggests that propionic acidemia predisposes patients to bilateral base infarcts in the basal ganglia, and possibly this is due to dysfunctional cytochrome C oxidase and also hyperammonemia, leading to accumulation of glutamine and or glutamate. Now, neonates may present in the first few days of life with decreased feeding, vomiting, lethargy, seizures, and hepatomegaly may be present. And patients with infantile or late onset forms may have a failure to thrive, developmental delay, seizures, spasticity, abnormal movements like dystonia, rigidity, and choreoathetosis. Now, the dietary treatment includes a protein-restricted diet, which is the cornerstone of treatment, a low-protein diet, L-carnitine supplementation, and also biotin supplementation will help in propionic acidemia. Now, let's move on to see our next disease, that is methylmalonic acidemia. Now, let's move on to see methylmalonic acidemia. And the methylmalonic acidemia, again, it's an autosomal recessive disease due to mutation in MUT gene leading to defective methylmalonyl-CoA mutase enzyme. Note that this methylmalonyl-CoA mutase is the one that converts methylmalonyl-CoA into succinyl-CoA. So, this is the genetic cause for methylmalonic acidemia. So, this is the inherited cause for methylmalonic acidemia. There can be acquired cause for methylmalonic acidemia, and that is because of vitamin B12 deficiency. Particularly, it is the 5-deoxyadenosylcobalamin deficiency. Now, out of these two, that is inherited cause and the acquired cause, mutations in MUT gene, which is coding for methylmalonyl-CoA mutase enzyme. It is the cause for 60% of methylmalonic acidemia cases. Now, methylmalonic acidemia can manifest itself differently depending on the following factors. If it is a total absence of enzyme, so the manifestation is different, which is written as MUT0, that is means MUT gene is totally kind of deleted. And if there is a reduction in the enzyme activity, that is MUT minus kind cases, where there is a decreased activity of an enzyme, or it can be defect in the synthesis of 5-deoxyadenosyl-cobalamin, that is vitamin B12 being decreased, or it can be defect in the cobalamin metabolism, which appears as both methylmalonic acidemia and also homocystinemia. Now, what are the clinical signs and symptoms in methylmalonic acidemia? Patients with methylmalonic acidemia can manifest with vomiting, dehydration, lethargy, seizures, recurrent infections can be seen. Progressive encephalopathies are some of the features of this particular condition. And the methylmalonic acidemia is due to dearrangement of deoxyadenosyl synthesis and cobalamin metabolism, may also have features of homocystinemia. So it can be combination of two things if it is a deoxyadenosyl-cobalamin enzyme deficiency. So that is methylmalonic acidemia along with homocystinemia, because cobalamin is also necessary for the conversion of homocysteine into methionine, and that reaction can be affected. That's why in cobalamin deficiency, as opposed to the inherited cause of methylmalonic acidemia, in cobalamin deficiency caused methylmalonic acidemia, you can see elevated methylmalonic acid along with homocystinemia. Now, the signs here is homocystinemia related signs along with like hypotonia, lethargy, failure to thrive, hepatosplenomegaly, monilial infections, chorioathetosis, dystonia, dysphagia, and dysarthria. So there are so many signs that are associated here with methylmalonic acidemia. Now, dietary treatment includes protein restricted diet, L-carnitine supplementation, and cyanocobalamin supplementation. So this is about methylmalonic acidemia. Now let's move on to see oxidation of very long chain fatty acids. Our diet, some proportion of very long chain fatty acids which have got carbons 22 or more. And also very long chain fatty acids are synthesized in our brain cells and also neurons by elongation process, because these very long chain fatty acids are required for sphingolipid formation, myelin synthesis and all that. Now, oxidation of very long chain fatty acids will be going on in two subcellular organelles. One is in the peroxisome and other is in the mitochondrial matrix. Before we get into the oxidation of very long chain fatty acid, let me briefly explain you what is actually the function of peroxisome. See the peroxisomal function. Peroxisomes are ubiquitous organelles in our cells. What they do is, they are involved in the oxidation of variety of molecules, that is, they are involved in the oxidation of VLCFA, very long chain fatty acid oxidation. They are involved in the oxidation of BCFA, BCFA means branched chain fatty acid oxidation. They are involved in the oxidation of ferritin, You know ferritin is a iron containing molecule, iron storage protein is ferritin. They are involved in the oxidation of ceruloplasmin. Ceruloplasmin is a copper containing protein. They are involved in the oxidation of purines. Purine oxidation is a uric acid formation. So purine oxidation is going on in the peroxisomes. Not only that, so their peroxisomes are also involved in synthesis of bile acids. Bile acid synthesis is going on in the peroxisome. Synthesis of plasmalogens. plasmalogen synthesis is going on in the peroxisome. So these are just to name few. These are some of the functions of peroxisomes, right? So peroxisomes, one more time, just for you to make sure you remember that. Peroxisomes are involved in oxidation. Initially I am saying the oxidative function of peroxisome. Oxidation of VLCFAs, oxidation of BCFA, branched chain fatty acid, oxidation of ferritin, oxidation of ceruloplasmin. Ceruloplasmin is for copper, ferritin is for iron, oxidation of purine into uric acid, and then I am explaining now I am giving you the synthesis function, synthesis of bile acids and synthesis of plasmalogens. All this is going on in the peroxisome. This is what is the function of peroxisome, some of the functions of peroxisome. And also peroxisomes are involved in detoxification process also, right? Now, as I said to you earlier, that oxidation of very long chain fatty acids which is going on in the peroxisome. So in fact, oxidation of very long chain fatty acids will be going on in two subcellular organelles. One is peroxisome. Initially it is in the peroxisome, and later it will continue in the mitochondria. So what happens here is a chain is shortened to short and or medium chain length in the peroxisome, and later it will be transferred to mitochondria for further beta oxidation process. Okay, so in this process of very long chain fatty acid oxidation, a hydrogen peroxide is the byproduct of VLCFA oxidation, and this hydrogen peroxide, it is neutralized by a catalase enzyme into water molecule. Catalase is the enzyme that neutralizes the hydrogen peroxide which is produced in the peroxisome by VLCFA oxidation and also by purine oxidation into uric acid formation. There is a generation of hydrogen peroxide. That's why peroxisomes, they will express highest concentration of catalase enzyme, so that hydrogen peroxide is neutralized into water and hydroxyl radicals formation is prevented. Now here is the figure. It will show you the oxidation of very long chain fatty acid, where VLCFA, it is activated into VLCFA-CoA, and this on the outer mitochondrial membrane, and the VLCFA-CoA, it is getting into the peroxisomal matrix, where it is oxidized into either short chain fatty acid length or medium chain fatty acid length. During this process, there will be release of acetyl-CoA molecules, and also more importantly, there will be release of hydrogen peroxide as a byproduct, and this hydrogen peroxide is taken care by catalase enzyme. So what will happen to acetyl-CoAs and short chain fatty acids and medium chain fatty acids, fatty acyl-CoAs that are released here. So acetyl-CoA, short chain fatty acyl-CoA and medium chain fatty acyl-CoA, all of them, they are taken out of peroxisome into the mitochondria by esterifying them with carnitine. So the carnitine is the one that helps in the transport of short and medium chain fatty acyl-CoAs out of peroxisomes, including acetyl-CoAs, out of peroxisome into the matrix of mitochondria for further oxidation. So once these molecules reach matrix of mitochondria, so acetyl-CoA will be reformed, SCFA, MCFAs are reformed, acetyl-CoA gets into the TCA cycle to undergo complete oxidation, and MCFAs and SCFAs, they will undergo beta oxidation to form acetyl-CoA, and acetyl-CoAs further will undergo TCA cycle. Like this, very long chain fatty acids are oxidized in two subcellular organelles. One is in the peroxisome, and later it is continued in the matrix of mitochondria. Now a disease that is associated with oxidation of very long chain fatty acid defect is Zellweger syndrome. Now the Zellweger syndrome is because of a mutation in a gene that is coding for peroxins, and the name of the gene that is coding for peroxins is PEX1 gene, and this defect lead to changes in the normal assembly and biogenesis of peroxisome. So there is a defect in the normal assembly and biogenesis of peroxisome, thereby peroxisomal function is altered here, and because of this there will be accumulation of very long chain fatty acids, branched chain fatty acid, because I told you function of peroxisome is oxidation of VLCFA, oxidation of BCFA, oxidation of ferritin, which is one of the function of peroxisome, oxidation of ceruloplasmin is a function of peroxisome, and purine degradation, function of peroxisome, bile acid synthesis, plasminogen synthesis. So all these are the functions of peroxisome. So there is a catabolic function and also there is an anabolic function. So when peroxisomes are defective here, so both catabolic functions are affected and anabolic functions are affected. That means VLCFA will accumulate because they are not oxidized. BCFA will accumulate, branched chain fatty acid accumulates because they are not oxidized. Ferritin will increase, ceruloplasmin will increase, and also there will be decrease in bile acid synthesis, plasminogen synthesis, and purine degradation will decrease. So because of all these metabolic changes, so there can be demyelination of the neurons, because peroxisomes are important for oxidation of very long chain fatty acids which are abundantly present in the neurons. So demyelination of neurons may lead to severe psychomotor retardation, profound hypotonia with depressed deep tendon reflexes, hearing and vision loss, and seizures. Along with that, patient will show hepatomegaly, high copper levels, high iron levels because of not degrading ferritin and ceruloplasmin, severe neurological defects, hypomyelination, craniofacial and skeletal malformation, including large anterior frontal nullae, hypoplastic supraorbital ridges, broad nasal bridges, micrognathia, deformed earlobes, and redundant nuchal skin folds. So there are so many signs and symptoms that are associated. Most of them are like neurological issues here. But most importantly, I would like to say here is to diagnose Zellweger syndrome rather than looking for clinical signs and symptoms. Of course, neurological signs and symptoms are much higher here, but we got to look for the laboratory features of Zellweger syndrome. Along with the neurological signs, the laboratory signs of Zellweger syndrome will help in the diagnosis process, and that is, we need to look for increase in VLCFA levels, increase in BCFA levels, that is increase in very long chain fatty acids, increase in branched chain fatty acid, increase in iron, increase in copper levels will help in the diagnosis process. So look for these things along with the neurological signs, and that will help in the diagnosis, and that is Zellweger syndrome, and that's because of mutation in PEX1 gene which is coding for peroxins, and peroxins are the proteins that help in normal assembly of peroxisomes. So that's about oxidation of very long chain fatty acids and oxidation of odd number carbon fatty acids, and also we have covered propionic acidemia, methylmalonic acidemia, and Zellweger syndrome. Thank you.